This is for Biology 223 Genetics. We have a different article today, article from a different journal. This is from PNAS, that's the Proceeding of the National Academies of Science of the United States. It's a journal put out by the National Academy. A National Academy is a body of scientists, they're academicians, they're academy members. It's prestigious. I'm not sure what the membership is, but this journal is put out and it's mediated by different ways where there can be a normal review submission or members of the academy can promote an article, still goes through some sort of review. And so we notice here, nor does it tell us who was contributed by the corresponding author, but it says who the reviewers were. Very often a review is blind, that is, people are chosen by the editors and the authors of the paper don't know who those reviewers are. So we say peer review and blind peer review. Some journals have a double blind system which doesn't really work, in which the reviewers don't even know who the author is. That's often, if the reviewers are specialists, then it's very hard for them not to know who the author is. That's not very common, but I've seen it. And sometimes there's pretty much open. eLife is a journal in which not only the reviewers are known, but the paper is published with the reviews of the reviewers and the author's response to the review. We haven't spoken much about the publishing process, but that's that people say it's a terrible system, but it's the best one we have. So a couple other things here. This journal covers all sciences, and so here that says genetics, so they put it in the category of genetics. They even cover sociology. Also, which may not be visible here, it says that it was downloaded at AUB on the date. So you can see that I downloaded this on December 15, 2019. Okay, this was published October 8th, 2019. And let's go through this. This is rather interesting to me that it was published in this journal. It's a good journal. It's not the most prestigious journal. And first thought that crossed my mind was, well, this is pretty important. Why isn't it published in a more prestigious journal, a more glamorous journal? And I think partly because of the experiments that would be demanded. If a journal like Cell would demand many, many experiments to nail down the mechanism in great detail, and perhaps the authors wanted this published without having to completely or to elucidate it as far as another journal might require, or they're in a hurry, or maybe it's not considered glamorous by the reviewers of some of those journals. So the nucleosome core particle remembers its position through DNA replication and RNA transcription. So remember, we have DNA is packaged in these coils, and there's histones, and histones are there. And so when DNA is transcribed or replicated, there's a replication bubble, a transcription bubble, and the DNA is separated into two strands. So the nucleosomes have to depart. They have to depart. They're pushed aside by helicases associated with the transcription elongation complex or the replication complex, and then they have to load back on. And so this tells us that the core particle there, the histone, maybe associated factors, remembers where they were through transcription. So when the bubble transcription or replication bubble machinery pushes, and the replication machinery or the transcription machinery pushes a nucleosome off the DNA, then they remember where to go. And this is an important issue in epigenesis, because the DNA, the epigenetic marks, are thought to exist not only by the easier to understand mechanisms of DNA being physically marked by things such as methylation of cytosine at CpG sites, but also by different modification, cetylation, and methylation of arginines on histone proteins. So let's go through this. Nucleosomes are the fundamental structural unit of chromatin. In addition to stabilizing the DNA polymer, nucleosomes are modified in ways, they're chemically modified in ways that reflect and affect gene expression. So we have special modifications of nucleosomes that occur in promoters, in gene bodies, at the ends of genes. So we have special modifications that are associated with transcriptionally quiescent DNA, with transcriptionally active DNA, with the beginning and the body and the end of genes. And these are sophisticated, and these can be, their modification is mechanisms by which they're modified, and the information that controls that is a very important topic. Modified in ways that reflect and affect gene expression in their vicinity. It has long been assumed that nucleosomes can transmit memory of gene expression through their covalent post-translational modifications. One point is that if the DNA is modified to show where the difference between heterochromatin and euchromatin actively transcribed and accessible to DNA, to transcription initiation or not, then after a round of replication and mitosis, or even after transcription, would that be erased? With the physical chemical marks on the DNA would still be there, like the 5-methylcytosine, but the histones, how would they find their way back and know where to go, and how is that maintained? One could imagine that it's primarily resides in the DNA modifications, and the histones find modified histones find it by that. But it seems that there's the assumption was that the histones know where they are. Okay, it's long been assumed, but it's an assumption. So it's an assumption without evidence, without convincing evidence. It has long been assumed that nucleosomes can transmit memory of gene expression through the covalent post-translational modifications, such as arginine methylation and arginine acetylation. An unproven, let's say, or an assumption without convincing evidence, unproven assumption in this model, which is essential to most models of epigenetic inheritance, not all but most, is that a nucleosome present at a locus reoccupies the same locus after DNA replication. Well, okay, first of all, we can assume that because it explains all sorts of things, but the question becomes, well, do we have any evidence for it? And by what mechanism would it happen? Can we consider a plausible mechanism by which a nucleosome that's displaced would know where to go back? If there's some machinery attached to the replication or transcriptional elongation complexes that take it ahead and put it back as they're going, okay. But one has to conceive of a mechanism, or it's hard to be convincing. We tested this assumption. So they tested this assumption experimentally by nucleating a synthetic chromatin domain in vivo. So they in vivo they made a bit of chromatin, a DNA, and with all the stuff necessary to do that, in which approximately 4 nucleosomes at an arbitrary locus were covalently labeled with biotin. Biotin, small molecule, biotin, then essential in humans through the diet. We don't synthesize it. It's essential coenzyme. The use of biotin is that there's an extremely tight interaction with streptavidin. So biotin-avidin interaction is essentially, once it forms, the off rate is so slow that we can use it to label things and detect molecules and single molecules even. We tracked the fate of labeled nucleosomes through DNA replication, established that nucleosomes present at a locus remembered. So what does that mean? They turned up there again. Doesn't mean they have memories of the same sense we do, but one has to use language that makes sense here. Remembered their position during DNA replication. The replication-associated histone chaperones DPB3 and MCM2 were essential for nucleosome position memory. So this is one of, so first was this statement of the general area, the gap in knowledge, which was essentially there's a model without supporting evidence, their method, and what they found. And here we have a finding that these histone chaperones were essential for nucleosome position memory. That acts to support their model, because it says that here's our model, and this model makes sense because if this model were true, then these other factors are involved, and we can specifically remove them and show that it doesn't happen without them. So that supports their model, as well as giving a detail explaining the mechanism. And in the absence of both DPB3 and MCM2 histone chaperone activity, nucleosomes did not remember the position. So in their presence, they saw that. In their absence, it didn't happen. So that supports their model. It's a very important piece of evidence. Using the same approach, and we can see here in their keywords, chromatin, epigenetics, Saccharomyces, DPP3, and MCM2, you see the choice of keywords, so we can suspect they use yeast, and that there are people who might be interested in these chaperones who are interested in this paper and its findings. Using the same approach, we tested the model that transcription results in retrograde transposition of nucleosomes along a transcription unit. So apparently there was a belief or assumption by people that when transcription occurred, that nucleosomes were displaced behind the elongating transcription complex, and they tested that. We found no evidence of retrograde transposition. The results suggest that nucleosomes have the capacity to transmit epigenetic memory across mitotic generations with exquisite spatial fidelity. Okay, so this has all sorts of interesting ideas here, something long-standing, and in some extent this is maybe like the Mesos and Stahl experiment for nucleosomes. Let's also go through the, so it has a nice introduction, it's quite accessible. Let's go through this. This journal requires a significant statement. So the authors, in addition to having to write an abstract, having to come up with a title and write an abstract, they also have to write a significance statement, which, unlike the abstract, is more an attempt to explain to the lay scientist why this is important, what is its significance. This is a very good thing. This is what we would like to be able to write after taking this course. After completing this course, we should be possible to write some of these on articles that are not too technically sophisticated. Chromatin modifications are broadly believed to influence gene expression. That's without doubt, and are thought to transmit epigenetic memory of gene expression across mitotic generations. Yes, and I had seen that belief, and the first thing that came to my mind was, okay, yes, but how? What mechanism? The best available evidence about the fate, there has to be some mechanism, there has to be some machinery, there has to be some way they can find what is the physical basis of their memory. The best available evidence about the fate of nucleosomes during DNA replication is at odds with this intuition. So the problem is the evidence didn't support it. This work studies the biochemistry, genetics, biochemistry, of chromatin replication in living cells, demonstrates that nucleosomes can in fact transmit epigenetic memory. Furthermore, contrary to published models, nucleosomes were not observed to translate locally during transcription. There was, they refer to it, there was some work with SV40, which is a small DNA virus that acts as a replicon in cell extracts, in which they saw, they did not see the same situation. So that was in cell extracts rather than living cells, and that's presumably the reason for that. Were not observed to translate locally during transcription. This work motivates a substantial reevaluation of published biochemical data about chromatin replication transcription. So they've opened up a major point that here it is, look, our intuition, we have a method to study this, and we see this, and it's in contrast with some previous published findings about how transcription works. And so the obvious question is, what are the details of the mechanism? How is it that a nucleosome can remember its position? How does it do that? And that's then for later study. But that to show that even in a journal is not front rank, glamorous, specialized, there's some major theoretical puzzles that come up and are, at least here, partially resolved.